wonderful. I'm just thrilled, delighted to be here to be able to welcome everyone today to what is now the fourth annual Barondes Lectureship. I have the great pleasure of being able to introduce our outstanding speaker, Rob Malenka, today. But first, I want to say a few words about the lectureship itself. This was established in 2017 to recognize and celebrate Sam Berendez' remarkable contributions to the department, to UCSF, to the field of psychiatry, and to neuroscience broadly. As you all know, Sam has been a true pioneer from the very beginning of his career. His early work in the laboratory laid the foundation for the pursuit of molecular mechanisms underlying serious mental illness. As the chair of psychiatry at UCSF here in 1986, beginning in 1986, he set his sights on identifying and recruiting the very best young physician scientists in the world, doubling down on the bet that cellular, molecular, and developmental neuroscience and human genetics would provide key mechanistic insights and provide an avenue to develop more rational and better and more effective treatments for the most severely affected patients. It is remarkable always to think about how prescient this vision was. While the road has been a long and winding one, no doubt much longer than Sam would have liked, we are now finally beginning to realize the promise that he recognized so early. Among Sam's many gifts, he has an unparalleled eye for talent and an equally remarkable persuasiveness. The physician scientists that he managed to bring to UCSF over the years, often with very modest means, is truly a who's who of psychiatric neuroscience and genetics. John Rubinstein, Mark von Zastro, Alison Dope, David Cox, Nelson Primer, Vika Sohal, Anna Malofsky, Dave Manoli, and today's speaker, Rob Malenka. Sam has always been a man ahead of his time, and not surprisingly, our discipline was not always ready to embrace him or to hear what he had to say. I remember when I was applying for residency, which was at the end of Sam's first term as chair, many psychiatry faculty around the country commented dismissively that UCSF had lost its way and hired a slime mold biologist to lead Langley Porter. The implication, of course, was that he could not possibly appreciate the humanity and complexity of psychiatric illness. They could simply not have been more wrong. Sam is a brilliant scientist, a leader in the field, a true mensch, gracious, generative, generous, and deeply committed to making the world around him a better place. It is a true honor and pleasure to celebrate and recognize his many contributions with the Barondes Lectureship. Now on to Dr. Malenka. Dr. Robert C. Malenka is the Nancy Friend Pritzker Professor of Psychiatry and Behavioral Sciences and Director of the Nancy Pritzker Laboratory in the Department of Psychiatry and Behavioral Sciences at Stanford. And he's Deputy Director of the Wu Tsai Neurosciences Institute. Rob graduated summa cum laude from Harvard College. I'm very impressed by that. I am. Oh no, I think it's pretty tough always. He received his MD and PhD in neuroscience in 1983 from Stanford, where he completed his residency in psychiatry, and then did 4 years of postdoctoral research here at UCSF with Roger Nickel, who wonderfully is here with us today. Then, in perhaps the only misstep in an otherwise flawless career, he elected to leave UCSF and return to Stanford, where he launched a truly spectacular program of research that has literally transformed molecular and systems neuroscience. He began his career studying plasticity, in particular, the long-term potentiation and long-term depression triggered by NMDA receptors in the hippocampus. He established that postsynaptic changes in calcium concentration triggered LTP and LTD, and he produced an astonishing run of seminal papers elaborating the underlying molecular mechanisms. Simultaneously, over 25 years ago, he recognized the importance of developing approaches that would connect the synaptic work with circuit level understanding of neural mechanisms of behavior. He entered the field of addiction research and quickly became a leader, publishing yet another series of now classic papers showing directly that drugs of abuse elicit plasticity at the synapse in the brain's reward circuitry. This idea that we all now take for granted that addiction involves pathological forms of synaptic plasticity dominates virtually all research in the field and is directly attributable to Rob and his findings. And if that were not enough for 2 careers, Rob has now turned his attention to advancing the understanding of the neural mechanisms mediating social behavior and is redefining this field, something that we'll hear about more today. For a mere mortal like me, reading Rob's CV is breathtaking. It would take the rest of the hour to cover comprehensively, so I will just touch on a handful of highlights. He is an elected member of the National Academy of Sciences, the National Academy of Medicine. He has mentored 84 postdoctoral fellows. His papers have been cited more than 100,000 times. And I also have to say that I love the fact that he only lists 281 publications. So within this body of work are a truly jaw-dropping number of senior author papers in Cell, Nature, Science, and Neuron. And while he has been prolific, 281 is nothing to sneeze at. What is even more impressive to me is the quality and depth of the work and the way in which in each of the major challenges that he's undertaken, he's produced multiple spectacularly elegant, rock solid, and truly seminal studies. So finally, I want to add that Rob has a true passion for advancing clinical psychiatry. While he's had unparalleled success studying synapses and circuits, he is as committed to the ultimate goal of leveraging scientific knowledge to improve the lives of those with serious mental illness as anyone I've ever met. It's one of the reasons that his selection as the 2022 Barondes Lecturer could not be more fitting. Rob, thank you so much for doing this. I know Sam is enormously appreciative, and it's wonderful to have you back. Get out of your way.
All right, are there any questions? Well, thank you, Matt. What I think Matt forgot to mention, which will be part of the beginning of my talk, is that I did spend, after my postdoc with Roger Nickel, 10 years on the faculty here at UCSF before I moved to Stanford in 1999. And so I thought I'd begin by telling you a little bit about my relationship with Sam Berendez, who I want to ask, does he deserve the credit or the blame for my career? Because he was the one who first gave me a lab, a faculty position, and the opportunity to establish my own independent research program. So I owe him an enormous debt for that. And I thought I'd share a few anecdotes about my early interactions with Sam. The first one was in 1988, where, the truth be told, I never applied for a faculty job. I was a postdoc with Roger. I wanted to stay in the Bay Area for my faculty job. There were only 2 choices, Stanford and UCSF. Stanford wasn't interested in me. And then I actually don't know how Sam found out about me. And I don't know if I reached out to you, Sam, or you reached out to me. But he said, what? I glowed. He says, some people are sitting in the audience. Sam's in the audience here, for those of you who are on Zoom. But somehow we started talking. It's 1988. And maybe Sam knew I wasn't willing to leave the Bay Area. So our first conversation was, okay, Rob, I want to give you a lab. You know, how about I'll give you 400 square feet at the VA and $100,000 startup. This is absolutely true. This is 100% true. And even back then, because I was a psychiatrist, this is 1988, and there wasn't a long history of what I would hope is somewhat sophisticated, rigorous neuroscience research in a psychiatry department. I was really worried about getting students and having a legitimacy at the UCSF powerhouse. I wanted to be treated seriously like an equal, which historically was not the case for scientists in psychiatry departments. So I said, you know, I don't think so. I need to be on campus. Campus was Parnassus campus back then. So I don't know if it was a week later, 2 weeks later, 3 weeks later. I didn't hear anything from Sam. So then he goes, well, this is absolutely true. How about 400 square foot lab at San Francisco General and $100,000 startup? And I thought about it and I said, no, the General is pretty far from Parnassus, and the Gallo Center was there, but I just don't think I can do that. So I said, I don't know. I don't think so. We need to be on campus. So I think Sam was desperate. He needed to hire somebody. I was the only game in town at the point. I think I may have been his first scientist hire. So he finally said, how about 400 square feet in the basement of LPPI and $100,000 startup? And I said, that will do. You are the man. I accept. So thank you for that, Sam. So then I started my lab in the basement of Langley Porter. And this is an absolutely true story. And I don't know if it was after a year of being on the faculty or 2 years or 3 years. At the time, I was trying to establish independence from Roger Nichol, who I'm going to mention in my next slide. Things were going pretty well. And so he brought me into his office. I don't know if Sam, you remember this. And he said, "What are you working on?" And I said, this is actually what I looked like maybe 25, 30 years ago. I had more hair. I had a mustache. I said, "I'm working on these phenomena of synaptic plasticity we call LTD and at the LTD, and they're pretty cool." And this is absolutely true. Sam looked me in the eye and he said, "LTP, schmelTP. What are you doing, Rob? Why aren't you working on molecules and proteins and genetics?" And he said, "Lectins are where it's at," because some of you know where that I'm joking about. And so I responded just like, "What am I going to do?" So I didn't give up studying synaptic plasticity. I don't know if I ever told you this, Sam, but even back then, and this is 1990, 91, I was thinking about molecular and genetic approaches to studying synaptic proteins. I was playing with viruses. Roger doesn't know this because we were not collaborating at this time. But in 1991, I was getting herpes viruses from collaborators at Harvard, dumping them onto cultured neurons. Roger's saying he knew all this. Dumping them on cultured neurons with the goal of trying to express dominant negative synaptic proteins, look at synaptic function. In 93 or 94, I got worms from Corey Bargman, and I was thinking of doing genetic screens, looking at neuromuscular junction synaptic transmission with a talented postdoc, Roselle Mulkey. Gave up on that a little bit too soon, but eventually the molecular revolution in neurobiology occurred. We had knockout mice, et cetera, and then I started using molecular manipulations of the sort that I think Sam would embrace. But more importantly, I want to say personally, thank you, Sam. He gave me a job. I think most importantly, he gave me freedom. Sam gave me no clinical responsibility. Eventually, I was an attending at a half-day clinic at the San Francisco VA for 7 or 8 years. But most importantly, he gave me a lab. He gave me freedom to just pursue my research objectives, and he always supported me. Every time I came back saying, "John Rubenstein is making $8,000 more a year than I am." True story. That upsets me. He matched John's enormous salary back then. When I said my 400 square foot lab in the basement of Langley Porter is hard to have 5 or 6 people, he gave me space on the 3rd floor of Langley Porter. And then things came from Stanford that were really hard to match, and I had to leave. So next, I want to say the same thing about Roger Nichol, and you can give him the credit or the blame. I was a postdoc with Roger for 4 years. I then became independent, and then for a variety of reasons, I think it was 93, 94, we decided to team up again. And I can honestly say my teaming up or marriage to Roger in the 90s was the most fun I have ever and will ever have in science. All right, and what Roger's saying is Sam facilitated that getting together. Okay, Sam did marital. I'm saying this for the people back on the Zoom. It wasn't that challenging, not like a real marriage, but it was enormous fun. It was enormous fun, and this is actually true. If you PubMed Nichol and Malenka, you'll find 75 publications. And as Matt, I think, alluded to, I would like to believe many of these are kind of citation classics in the synaptic plasticity field. So that's my history. I owe an enormous debt to my mentors here at UCSF. I still miss the place, as Matt State knows. And who knows? Maybe I have another decade, and who knows? Maybe I'll be back here one day. Who knows? Anyhow, so I'm told I have till 4:30. I'm not going to talk for another hour and 15 minutes, but I am probably going to talk for another hour. Sit back and enjoy the ride. I'm going to try to tell a scientific journey about a little bit about how I ended up going from studying synapses to empathy in mice, which is the last part of my talk. So let me begin. I began my career, as I mentioned, working with Roger Nichol, who introduced me topic of synaptic plasticity. That is these phenomena of LTP and LTD. My motivation is really described in the caption here. It was the assumption, the belief that all forms of experience-dependent plasticity, whether it be learning and memory, whether it be responses to a drug, whether it was due to a stressful event, that all experiences impact brain activity, neural circuits in the brain, and a major mechanism by which that experience influences circuitry and thereby influences thoughts, feelings, and behaviors must involve changes in synaptic communication and synaptic strength. So I convinced myself for the first, and I still believe this, for the first 30 years of my career, that understanding the detailed molecular mechanisms by which synapses, and these are mostly glutamatergic excitatory synapses, how they're modified was just gonna be at some point of fundamental importance for not only understanding how the brain worked, but most importantly for my passion, as Matt alluded to, understanding the pathophysiology of severe mental illnesses. And by doing so, we will finally develop better treatments. And like most of us in the field of academic psychiatry, it isn't happening nearly as rapidly as I had hoped 40 years ago. And so Roger and I had a good run. These are just some of the reviews we wrote in prominent journals. We published a lot of papers together. Enough said, and I continued to work on this with Tom Sudhoff when I moved to Stanford, although truth be told, for a variety of reasons, mostly that it's hard to attract students and postdocs interested in this topic, I'm really no longer working on the molecular mechanisms of synaptic function and plasticity. So basically in the late 80s, when I was still a postdoc with Roger, into the early 90s, I started getting worried. We were having a lot of fun studying mechanisms of LTP and LTD. It was a hot topic. I was making a good living. But I actually was intellectually concerned about the question of whether these phenomena actually happen in the awake behaving animal's brain and actually play a role in any form of experience-dependent plasticity. And the reason I was concerned is if any of you actually know the long history of research of LTP in the hippocampus, it had actually proved very difficult to design experiments that definitively proved that synaptic weight changes on the main output cells of the hippocampus, CA1 pyramidal neurons, that synaptic weight changes actually were necessary for real learning and memory. And the challenge was probably that the hippocampus uses a very sparse code to encode new experiences, new memories. So just if you ran the mouse through a maze, if you ran it through fear conditioning, actually, especially remember, this is in the late 80s, early 90s, when we didn't have tools to potentially identify activated ensembles of neurons in response to some experience. I just figured it was like finding a needle in a haystack. You were never going to be able to find synapses and look for weight changes in response to an experience. So I really, beginning as a senior postdoc with Roger, when I started my lab, I was looking for brain areas and synapses and experiences to give the animal where that might be a little bit easier. And truth be told is I was forced to give a series of lectures to graduate students. I don't know when this was. Maybe I was an assistant professor by then in 90 or 91 on the limbic system with Mike Stryker. And I started getting into the literature on the limbic system, and I started reading the literature on the neurobiology of drug addiction. And even back then, some of the investigators in that field were talking about the idea. So truth be told, it wasn't my original idea. They were talking about the idea that plasticity mechanisms in classic mesolimbic reward circuitry, which is shown in this slide, in the VTA, which is the home of dopamine neurons, as most of you know, their projections to the nucleus accumbens, their projections to the prefrontal cortex, that some forms of plasticity in these neurons might be important for the behavioral adaptations in response to administration of drugs of abuse. And the major evidence for that, I'm going into probably more detail than you want to know, but just to let you know, was that there were papers reporting that an NMDA receptor antagonist infused into the VTA, and as hopefully most of you neuroscientists know, NMDA receptors are key triggers for synaptic plasticity, that infusion of NMDA antagonist into the ventral tegmental area could prevent some of the behavioral adaptations in response to psychostimulants like cocaine and amphetamine. And then I started reading the literature and I realized, with all due respect to my addiction colleagues, these people had no idea how to study synaptic biology. They were mostly behavioral pharmacologists or neurochemists by training. So I saw there was an opportunity there. And so I just did what at the time I thought were obvious experiments. The only mistake I made is the first set of experiments I did initially at UCSF, and then I carried them forward at Stanford, was just to confirm that excitatory synapses on the main cell types of dopamine neurons in the VTA and medium spiny neurons in the nucleus accumbens could express some form of LTP or LTD. And it's much harder to generate those phenomena. And then I should also mention, which all of you know, the other major interest in this classic mesocortical limbic reward circuitry is, hopefully for obvious reasons, these brain areas, these connections, these circuits very likely play an important pathophysiological role in major mental illnesses such as addiction and depression, probably obsessive compulsive disorder, et cetera. Okay, so then in the early 2000s, or actually the late 90s when the work was actually done, I recruited some postdocs and we did a series of experiments where we gave animals drugs of abuse, usually cocaine, because that was the popular drug back then. And we gave the animals cocaine. We then cut slices of the VTA or accumbens, and lo and behold, we were able to detect synaptic weight changes. And I think this particular paper was the most important one, because in this paper, which was published in Neuron, again now 20 years ago, which Neuron actually wanted to reject initially, but I convinced even back then I was good at convincing editors to pay attention to my work. I convinced the editors that it might be important. What this paper showed is that several different classes of drugs of abuse caused synaptic weight changes as measured by this ratio, the AMPA-NMDA ratio, that they all did cause the synaptic weight change. And that sort of gave greater credence to this idea that synaptic plasticity mechanisms actually could be maladaptively usurped by drugs of abuse. And I would like to believe, although it may be a fantasy, that this work really did change the field of addiction research and started getting people first looking at synaptic changes in more sophisticated ways, then started the whole circuit revolution, not only in addiction research, but it's happened in all of neuroscience. Okay, so I was now thinking about reward circuitry, and I was starting to read about it and think about it. And I was really influenced by finding this very simple hypothesis first proposed by the American psychologist Thorndike, which he called his law of effect, which is intuitively obvious. And that is that all of us increase the behaviors that we manifest that lead to some reinforcement or reward. And we avoid, in general, the behaviors that lead to pain or punishment or an aversive experience. And I would argue that when all is said and done, most animal behavior, most human behavior can actually be explained by this very simple postulate. We're just much more complicated species because we have this huge cortex, which complicates things and allows us to put off rewards by 20 years, like going to medical school and becoming a professor. And it allows us to put up, enough said. So I really became interested in this question of, can you actually discern the circuits and the mechanisms that actually tell you an experience is rewarding or tell you that an experience is aversive? So now we're getting into the realm of, I hate to say it, psychology. So we started working on VTA dopamine neurons. We published, and this is a little bit of a show-off slide. We published a series of papers using now the modern tools of optogenetics. For this paper, viral tracing methods in collaboration with Lichun Luo to define the heterogeneity of VTA dopamine neurons to a much greater extent, which is now, I'm actually pleased to say, most people in the dopamine field now have accepted. At the time we started publishing this work, there was the dopamine field, and it still is, it was dominated by sort of thinking around computational models. Most importantly, this idea that dopamine neurons encode, my favorite word, encode reward prediction error. And while that's certainly likely the case, it seemed to me it was intuitively obvious that dopamine neurons in any neuromodulatory system must do a lot of different things. It didn't evolve to do this one single computational task because computational neuroscientists liked how simple it made everything. So we started looking into that. We started looking at synaptic adaptations in the ventral striatum and the nucleus accumbens, trying to connect them up to mouse models of behavioral changes related to amotivation and anhedonia and depression. I will admit, unfortunately, we have not followed up on these papers, but at least it said you could attack these problems using sort of, with a synaptic perspective. I teamed up with Tom Sudoff. We did some analyses of mouse models of autism. So, the lab was going along pretty well. But I was still struck by this question of what is rewarding or aversive. By this point, and this is now like 12 or 13 years ago, the addiction field had become actually pretty competitive. And again, to be a little whatever obnoxious or narcissistic, it was competitive because several postdocs that either Roger and I had trained together or I trained on my own were now in the addiction field and doing lots of the experiments I had hoped to do myself, and I couldn't keep up. So I started thinking about other experiences and stimuli I could study that likely involved reward circuitry and that were equally rewarding or aversive, but there was room to maneuver as a scientist. So actually, we did quite a bit of work on feeding. It's just none of it has ever been published because the results were just not reliable enough. But then about whatever it was, 13, 14, 15 years, a postdoc showed up in my lab, Gould Dolan, who's now a professor at Johns Hopkins, and we started talking about studying social behaviors with the obvious intuition that on a good day, when you're in a good mood, social interactions are highly reinforcing and are evolutionarily adaptive. So it just seemed intuitively obvious that if there is this circuitry in the brain telling us that something is reinforcing and rewarding, a social experience must engage that circuitry somehow. Furthermore, social interactions can be highly aversive. And anybody who's been married or in a serious relationship knows your partner can go from changing to be highly rewarding or highly aversive in about 10 seconds, sometimes 2 seconds. And as neuroscientists, we know that does not happen magically. That has to be a neural mechanism by which that occurs. There has to be some change in neural activity that's telling you your wife is now your ex-wife. So we started thinking about how do we attack the complexity of social behaviors within this context of reward circuitry. So now in the next 45 minutes, I'm going to take you on my scientific journey, which actually has led me to studying empathy in mice. So it began by publishing this paper, which I'm not going to go through any of the details. It was published now almost 10 years ago. It's a very complicated mechanistic hypothesis. All the data were consistent with this hypothesis. And it was basically that during an adaptive, what I'm going to call pro-social interaction between two conspecific mice of the same sex, that oxytocin is released in a key node of the reward circuitry, the nucleus accumbens. And oxytocin causes the release of serotonin from serotonin inputs from one of the major serotonergic nuclei in the brain, the dorsal raphe. And this release of serotonin in the nucleus accumbens is necessary for the rewarding component of social interactions. And again, I'm not providing any evidence for this hypothesis, how we generate it to you. All I want you to know is that this hypothesis predicted that the downstream mediator or a necessary component of a pro-social rewarding interaction had to be serotonin release in the nucleus accumbens. And let me give Roger Nickell another compliment. I find myself even now, 20 years after working with Roger, I still find myself saying things to my postdocs that Roger used to say all the time. And one of my favorite lessons from Roger was, if you have a hypothesis which can be disproved by an experiment, you are morally bound to do that experiment. You must do that experiment. You must test your hypothesis. And those are the experiments which you're scared about. Because if they don't work out, it means all your conclusions, all your work, you're wrong, and you have to rebase. But so I was trained by Roger. So the obvious experiment was manipulate serotonin release in the nucleus accumbens. And if this complex hypothesis is true, manipulating serotonin release in the nucleus accumbens must influence prosocial behaviors. So Jessica Walsh joined my lab now, 8 years ago, and decided to do these experiments. This is published. I'm going to go through these data pretty quickly. I probably have presented them here at UCSF a few years ago. So she did what I hope to those of you who are neuroscientists in the audience is the relatively obvious experiments. We decided to optogenetically manipulate serotonin release in the nucleus accumbens by expressing transgenes in serotonin neurons. And all of these experiments were performed and analyzed blindly. So here's the protocol. You express channelrhodopsin in a driver line, the Circuitry driver line, so you can express transgenes specifically in serotonin neurons in the dorsal raphe. You shine the light in the nucleus accumbens, so you can either activate serotonergic inputs in the nucleus accumbens, or using a different optogenetic reagent, you can inhibit input activity and presumably inhibit serotonin release. And then you run the animals through a bunch of behavioral assays. With the awesome power of optogenetics being, you can look at the behavior of the animals with the light off, the control behavior, and then turn it on. And the behavior under the influence of increased serotonin active release or decreased release. And remarkably, we got very reliable and robust results. This shows the individual animals and the variability. This shows the mean. And what this shows you is that activation of serotonin inputs increased interaction with a young conspecific juvenile, the juvenile interaction assay. So these are very simple behavioral assays. You just ask your subject mouse, how much time do you want to hang out with your little buddy in a non-aggressive, in quotes, pro-social manner? And then the other assay Jessica did is the standard 3-chamber social preference assay, where you have a 3-chambered house. On the outer chambers, you have a cup with a juvenile conspecific mouse. In the other cup, it's either empty or you have a toy mouse. And you ask the mouse, where do you want to hang out? Do you want to hang out with your little buddy or not? So in both of these assays, when we presumably release serotonin in the accumbens, we promote sociability. And remarkably, when we inhibit serotonin activity and presumably release in the accumbens, we reduce these sociability assays. So we have bidirectional control over these complex behaviors. And again, you can see the data. I hope it's right. I mean, it's pretty robust, but I think it's right because I'm now going to tell you another 40 minutes of data that all support this simple hypothesis. Okay, so then we decided to look at an autism mouse model, both because we're interested in pathophysiology and also, truth be told, I'm saying this in case there are any trainees in the audience, it's easier to publish the paper in today's world if you connect it up to a prominent neuropsychiatric disorder. We chose a mouse model of the 16p11.2 deletion syndrome, primarily because at the time, the head of the Simons Foundation, Jerry Fishbach, told me that's the mouse model I should study. And I was hoping to get money from the Simons Foundation. I said, whatever you say, Jerry. But more importantly, the truth is that colleagues at Stanford, Thomas Portman, who was a postdoc with Carl Dolmach, had made a conditional knockout of the syntenic region in the mouse chromosome. So we had the power of a conditional knockout, which we wanted to leverage. And hopefully it's obvious why having a conditional knockout. So the first experiment just look at these data, is take the conditional knockout of the syntenic region of 16p11.2 and either cross it with a Circuitry driver line. So she's genetically deleting this chromosomal segment only from serotonin neurons. And in a different population of Circuitry mice, she injected a Cre virus into the dorsal raphe, which will genetically delete this segment from all dorsal raphe neurons. And what you see is with either manipulation, she got a pretty robust deficit in both the juvenile interaction task. Whoops, sorry about that. Um, here's one set of data. Here's the other set of data in both assays. And obviously the control mice. We did a bunch of control behaviors that I'm not going to be able to tell you about, other than what we find remarkable about the serotonin finding is it doesn't seem to influence the animal's approach to other appetitive objects in its environment, like appetitive food objects, which is very different than what dopamine does in the nucleus accumbens. Okay, we then did a little bit of slice physiology. The only take-home message from this is that if you genetically delete this chromosomal segment from serotonin neurons in the dorsal raphe, they show some physiological changes. Their excitability is reduced. They fire off less action potentials. Their mini EPSC amplitude is changed. We haven't pursued this mechanistically at all. I don't know which genes that we deleted contribute to this, because again, the goal of this study was not that kind of mechanistic insight. It was to see whether a deficit in serotonin release in the accumbens might be a contributor to the sociability deficits in this mouse model of autism. And this just said deletion of this segment did cause physiological changes. We then made an attempt, our first attempt, we're much better at this technique now, to do what's known as fiber photometry. So in this experiment, we express a calcium indicator called GCAMP, which the changes in fluorescence of which correlates with the level of activity in which it's expressed. We then put a fiber optic into the dorsal raphe so we can measure the total fluorescence from GCAMP, which is a very coarse measure of the total neuronal ensemble activity of the neurons in which the GCAMP is expressed. It's expressed in dorsal raphe serotonin neurons. And what this shows you is in the autism mice, the amount of activity that you detect during a social interaction, time zero is when the subject mouse is just approaching the juvenile, is significantly less than in our wild type Circuitry mice. So again, it just said that these physiological changes in serotonin dorsal raphe neurons actually also have an effect on the activity of these neurons during a social interaction. So that allowed us to hypothesize that in these autism mouse model, there is, during a social interaction, there is likely a reduction in the amount of serotonin released in the accumbens. If that's the case, we reason we should be able to rescue the sociability deficits in this mouse model by releasing serotonin optogenetically. That's what's shown here. And you can see the results are just actually astonishingly robust. Those of you who know me can imagine how I threatened the postdoc who was giving me these results, because I just said, you better be right, because it's your career if you're wrong. But again, I'm going to give you a lot of data that makes me believe these were probably correct. So then we were interested in what receptor of serotonin might be necessary for these effects of serotonin release in the accumbens. I forgot to mention in the early work, the original paper from Gould-Dolan, we had found that a serotonin 1B receptor antagonist infused into the nucleus accumbens prevented or reduced our measurement of social reward during a pro-social interaction. So the obvious experiment was to optogenetically activate serotonin neurons and then infuse either saline or a 1B receptor antagonist into the nucleus accumbens and see what would happen. And here are the results. So all of these animals are the autism mouse model. All of them have channel rhodopsin in their serotonin neurons. We activate the somas of these serotonin neurons. The black lines show the animals that got saline into their accumbens, an independent replication of this robust rescue of the sociability deficits. But when we infused a 1B antagonist into the accumbens, we completely block the effects. We then actually tested whether a 1B agonist infused directly into the nucleus accumbens might also rescue the sociability deficits. And that's what this shows. There's a reason I'm telling you this that will become apparent in a few minutes. So the summary of this first sojourn into serotonin is that bidirectional modulation of serotonin release in the accumbens bidirectionally influences these prosocial behaviors, that a mouse model in which we delete a chromosomal segment implicated in one copy number variation associated with autism spectrum disorder has effects on the physiology of the serotonin neurons, and that we can rescue the sociability deficits in this mouse model by optogenetically releasing serotonin in the nucleus accumbens. Okay, so simultaneously, while this project was going on optogenetic manipulations of serotonin, another postdoc joined my lab, who again indirectly is related to me and Roger Nickel. His name's Boris Heifetz. He was an MD, PhD anesthesiologist at Stanford who was finishing his residency and wanted to do an additional postdoc. He did his PhD with a guy named Pablo Castillo, who was a postdoc with me and Roger. So, you know, it all comes back. And it turned out that both Boris and I had a real interest in using drugs as powerful probes of brain function. So why do I think drugs are powerful probes? It's because drugs have molecular targets. If you can figure out the molecular target, you can use the modern tools of neuroscience, conditional knockout mice, circuit manipulations, to start really figuring out how that drug, by interacting with its molecular target in specific cell types, modulates specific circuitry to mediate the behavioral effects of that drug that you're interested in. Furthermore, I'm interested in using drugs as probes of nervous system function because drugs like Mdma, which is also known by the street name of ecstasy or Molly, can be given either illegally or legally to human subjects. So you can actually you can at least talk to the human subjects and ask them what their experiences are on the drug. And in today's world, you can do what you can do in human subjects. You can give the drugs, you can do task-evoked or resting state functional magnetic resonance imaging, you can do EEG measurements, and you can try to build up connections between the more mechanistic mouse work and the human work. Now, Mdma is particularly interesting because, as many of you hopefully know, it has powerful effects on social interactions in human beings. It's pro-social. Some people even think it's empathogenic. And if time permits, which it may not, I'm going to show you that Mdma actually enhances mouse empathy in a very simple behavioral model my lab has been working on. So I went public with this interest now 6 years ago in a commentary from Cell. I actually said, okay, here's why I think studying Mdma is important for understanding one of the most important components of the human experience, that is social interactions. And to get on my high horse, I actually believe this. I believe our world is lacking so enormously in empathy and compassion. And I can't do anything about it other than perhaps by being a neuroscientist who studies these phenomena, maybe someday understanding the neurobiology of pro-social, non-aggressive interactions, even empathy, maybe the political leaders of the world will pay attention to that, and kumbaya, the world will be a better place. So that was part of the motivation. The other reason Boris and I were interested in this substance is, as many of you know, there's a hysterical excitement in the academic psychiatry community about the therapeutic potential of the so-called psychedelics. Mdma is not really a classic hallucinogen, but nevertheless, Rick Dobling and the people at MAPS deserve enormous credit for battling the battle, getting permission to study Mdma in a phase 3 trial in post-traumatic stress disorder, where they're getting pretty remarkable results. And that opened the door to now studying other drugs like psilocybin. And so, again, because I actually care more about the future of academic psychiatry than I did as a young pup when Sam hired me, because I see the lack of advances, at least the lack of advances that I had hoped would occur, I figure I got to do something in my last few years. So, and I don't want the field to go down the path, how to say this nicely, that it has historically, of not doing the best research on the drugs we give patients and making up simplistic hypotheses about pathophysiology of complex mental illnesses. I don't think that's helpful. I think it's actually counterproductive. And I'd like to do everything I can to help the field avoid that over the next 10 or 20 or 30 years. And that state is one of the reasons UCSF is at the forefront of preventing that from happening. Okay, the other reason I was interested in Mdma is, guess what? its major known neurochemical or molecular effect is to release serotonin because it interacts with the serotonin transporter. So it just all made sense. It also turns out it's been reported to influence oxytocin levels. It also interacts with the dopamine transporter and causes dopamine release with much lower affinity. Okay, this is also published. Although I have 30 minutes left, so I hope everybody take a breath. I have another 30 minutes to go. Hopefully you're with me so far, or if not, that's fine. All right. No, I'm not going to bore you that much. Okay, so Boris just he used the simple 3-chamber assay. He first did a dose response showing that 3 mg per kg of MDMA had a little bit of effect in social preference. These higher doses, which I want you to remember, 7.5 mg per kg versus 15 mg per kg, had a pretty robust effect in enhancing. It just froze on me. What do I do? Everybody take a breath. Smoke them if you got them. Oh God, restart. Yeah, take your Molly. No, just joking. Is it back? I'm sorry. This computer has never. UCSF did something to me. What do you think? Should we restart it? Sorry about this. I literally just gave this exact talk a few weeks ago with the exact same computer, and it worked. There we go. All right. I think it's John Rubinstein's fault. Anyhow, this is just the way Boris quantified the data. It's not important. This is a cute experiment where in this column, he just gave the MDMA to the mouse under the cup, maybe a little bit of an effect. In this column, he only gave the animal that's freely exploring MDMA and a slightly clearer effect. In this experiment, he gave both animals under the cup and the animal exploring MDMA, and he got a very robust effect. And to anthropomorphize mouse social preference in this simplistic assay, this is exactly what happens to human beings when they're taking MDMA at a party or a rave. There's a big difference if you're interacting with somebody who's on MDMA or not. So I am told. If it's frozen again. I apologize. I literally have used this computer many, many times. Thank you for trying to help me here. What else can I tell you? Maybe I should not use the pointer. Maybe let me think it's the pointer. All right, anyhow. Okay, so then the one thing Boris and I were worried about and interested in about MDMA is it is an amphetamine derivative. So it has psychostimulant properties. And therefore, like methamphetamine, dextroamphetamine, cocaine, by definition, it has abuse liability. And some of you know, back in the 90s, there was this hysterical concern. It's what led MDMA to become a Schedule I drug from a Hopkins professor who will remain nameless, who reported that MDMA in monkeys caused dramatic toxicity to neuromodulatory inputs. And it may be true at enormously high doses, but independent of that, it clearly has abuse liability. And we were concerned about that, and we wanted to understand that, both mechanistically, but also because it is going to probably eventually get FDA approval. And while I don't think MDMA is ever going to cause an epidemic like fentanyl did in the opioid epidemic, there is no doubt that once MDMA gets approved, there's going to be an enormous amount of off-label use. It's going to be more available recreationally, and there's going to be a lot of inappropriate use, and something bad is going to happen. So I believe if we can understand mechanistically how MDMA works, we can make a better MDMA. We can make one that has the pro-social, perhaps even empathogenic effects, and reduce its abuse liability. So Boris next did a dose response, doing two very simple assays, that the behavioral assays that correlate with abuse liability. One is just locomotion. All drugs of abuse with addictive liability cause an increase in locomotion in the animals. Cocaine, opiates, alcohol, nicotine. And what this shows is that 7.5 mg per kg that does cause a sociability enhancement in this assay we were using, doesn't cause an increase in locomotion. 15 mg per kg does. More importantly, he did a classic condition place preference assay where you give the animal the drug. In one context, you give them saline, in the other, and you ask the animal a day or two later, where do you want to hang out? And all rewarding experiences, all drugs of abuse that are reinforcing, cause a condition place preference. And what this shows is that 7.5 mg per kg does not, but the higher dose does. So we thought we had a dose that we could look at only the social effects, and then a higher dose that had both social effects and effects in these assays associated with rewarding properties, really. So as I mentioned, MDMA interacts. Its highest affinity target is the serotonin transporter. So we asked the obvious question of, if we prevent MDMA from interacting with the serotonin transporter, in this case using the drug citalopram, do we block the effects of MDMA? And that's what's shown here. The control animals that got saline with MDMA show this enhancement of sociability. Animals that got the antagonist to the serotonin transporter do not. Took a while. We then got a conditional knockout of the serotonin transporter. We, for a variety of reasons, I think Boris injected a Cree virus in the dorsal raphe, didn't cross it with the Circuitry. So now he's eliminating the serotonin transporter from serotonin neurons. And on average, in the heterozygotes and the homozygotes, he blocks the effects of MDMA on in this social preference assay. It's not the most convincing data. I'll show you other data later. He does not block the effects on condition place preference. So that says the interaction of MDMA with the serotonin transporter is necessary for the pro-social effects of MDMA, but not for its effects in condition place preference and its abuse liability. So then the next question was, is MDMA promoting social preference in the 3-chamber assay because of enhancing release in nucleus accumbens? All the other manipulations, we were just giving the drugs IP. So what this shows is that if you infuse, this is a different cohort of animals. If he gives MDMA IP, he gets noisy results, but on average, there's an enhancement in social preference. If he infuses MDMA directly into the nucleus accumbens, every single animal shows this social preference. The result is even stronger. So that says that MDMA action only in the accumbens is sufficient for this pro-social effect. This is the converse experiment where he gives the MDMA IP, so it's bathing the whole brain, but now he infuses either saline or citalopram, this antagonist to the transporter, into the nucleus accumbens. And when he prevents, in theory, pharmacologically, so this is old-fashioned behavioral pharmacology, although I still think there's a place for that if it's the right experiment to do, that on average, putting a drug into the accumbens that in theory prevents MDMA from interacting with the serotonin transporter, prevents its enhancement of sociability in the social preference task. And importantly, when he does these manipulations with a higher dose of MDMA, that is, he infuses citalopram into the nucleus accumbens, he doesn't block the condition place preference caused by MDMA. And just to show that the cannula were in the right place, if he infuses a dopamine receptor antagonist into the nucleus accumbens, on average, he can block the condition place preference elicited by MDMA. So together, this says that MDMA action in the nucleus accumbens, presumably interactions with the serotonin transporter, enhancing serotonin release, I'm going to show you proof of that in a few slides, is the necessary mediator of these pro-social effects. Does this have anything to do with therapeutic efficacy of MDMA? What Jess Walsh, who was still in the lab, did, she then studied the effects of MDMA actually in 6 different mouse models of autism spectrum disorder. This just shows 3 of them. The details aren't important. The bottom line is in every mouse model, MDMA promoted the sociability of these mouse models that in our assays express sociability deficits. So I hope I've answered this question. which is a phenomenological question, not a mechanistically driven question. Does serotonin release in the accumbens promote a pro-social behavior? And we think it does. This is just the final experiment I'm going to show you. Is the effects of MDMA in the nucleus accumbens, which causes the release of serotonin, also mediated or require activation of the serotonin 1B receptors in the nucleus accumbens? This is, again, another behavioral pharmacology experiment. If he gives MDMA IP and infuses a 1B receptor antagonist into the accumbens, he can block the prosocial effects of MDMA. We actually did some synaptic physiology, which I'm not going to talk about. So in addition to studying MDMA, Jess looked at a specific 1B agonist and its effects in these mouse models of autism. And again, the story is that in every one of these mouse models of autism, the 1B agonist seemed to enhance the social behavior of these mice, the prosocial behavior. Why am I emphasizing this so much? I'm not doing this anymore because I need to publish more papers in prominent journals. I'm doing this because I want to have an impact on human health. And I'm pleased to say that based on this work, there are 2 different biotechs pursuing these findings. Now, will they succeed? The chances are low, but this is an example of going directly from mouse to man, which a lot of people think you can't do, and maybe you can. But a biotech, I found it with Carl Deiseroff, called MapLight, which is going quite well, is actually about to dose individuals with autism spectrum disorder with a drug I'm not allowed to say the name of. We call it ML004, but it's based on our understanding that 1B agonist is important, 1B receptors are important. So we'll see if it has any influence in autism spectrum disorder. Another company for which I'm the chair of the SAB, it's one of the 2 major psychedelic companies. To be honest, completely based on my suggestion, are going to be pursuing an enantiomer of MDMA called the MDMA that MAPS and everybody is studying is a racemic mixture of RS-MDMA. It turns out if you look in the literature, R-MDMA has a much higher affinity for the serotonin transporter and a very low affinity for the dopamine transporter. So the reasoning was maybe R-MDMA might still have some of the pro-social, in quotes, therapeutic effect and have much reduced abuse liability. And they're pursuing this. And there's a long way to go, but they're actually dosing human subjects now in Switzerland in their phase 1 study. Okay, so, all right, I probably won't get to the empathy because I have so we're looking at serotonin. Another postdoc just arrived in the lab 3 years ago named Matt Pomerantz, and he came from an addiction background. So to bring this kind of work full circle, he was interested in the question of whether this change, serotonin release in the accumbens, or deficits in that might contribute to sociability deficits in other neuropsychiatric disorders. And as many of you certainly know, the major problem in the treatment of addiction is that it's a chronic relapsing disorder. And when people go through abstinence, they're abstinent for weeks, they very commonly go through this syndrome that George Koob has called hypercatipia. I won't tell you the etymology of that word. And this hypercatipia, these motivational deficit syndrome symptoms that addicts often go through, whether they're going through protracted withdrawal from alcohol, from opiates, from psychostimulants. There's a lot of overlap with depression, although it's not identical to depression, but people exhibit anhedonia, anxiety. Most importantly, people report social avoidance and isolation. And that's the last thing you want an addict to be experiencing, because that means they don't come to their therapy sessions. They don't get the social support they need to prevent their relapse. I'm going to skip that. So Matt decided to develop an animal model to probe the sociability deficits, in this case during protracted morphine withdrawal. So here's what he developed. He first tests the animals in these social behavior assays you're familiar with now. He gives them escalating doses of morphine. They show condition, place, preference, et cetera. He then withdraws them for 3 weeks, and then he tests them again. And all you need to know is, again, you get profound sociability deficits. mean, look at the raw data. Almost every single animal. And remember, you always have a baseline measurement and a post-withdrawal measurement. And obviously, everything is done blindly. We have saline control animals, et cetera. So during protracted opioid withdrawal, there is a sociability deficit. So how do we attack mechanism? I don't have time to really go into this in any extent. Matt decided to look at the role of kappa opioid receptors. There were several reasons for this. The first reason, not shown in this slide, is that there's an extensive literature, behavioral pharmacology literature and larger literature that has reported over the decades that kappa receptors in reward circuitry are very important for aversive behaviors and for changes in motivational behaviors elicited by stress in certain models of depression. And there's a lot more literature that I don't have time to review. Of equal importance, NIDA has labeled kappa opioid receptor antagonist as one of their top potential drug targets or drugs that might prove promising for the treatment of addiction. And then your very own Andrew Crystal here at UCSF published a paper in Nature Medicine reporting that a kappa receptor antagonist was an effective treatment for anhedonia and that it influenced activity in the human nucleus accumbens. So for all of these reasons, Matt did the following experiment. He decided to ask whether infusing a kappa receptor antagonist, again, a behavioral pharmacology experiment, we'll get more sophisticated by the end of this talk. If he infused the antagonist into the accumbens, does he block or prevent the sociability deficits? Now, he wanted to do within animal control. And the problem with NorBNI is it's kind of irreversible. Once you put it into the brain, it sticks around and influences kappa receptors for days or weeks. So he did the following experiment. He took all of his animals, gave them morphine, he withdrew them. He did baseline sociability deficits. All the animals showed the sociability deficits. Then half the animals got saline injections into the accumbens and half got NorBNI. You can see the animals that received saline still show the sociability deficits. So they're stable. You can test them every day for a few days. The animals that got NorBNI, that blocked kappa receptors in the accumbens, the sociability deficits were essentially completely reversed. Where is the dynorphin is the endogenous ligand for kappa receptors. Where's it coming from? We assumed it was coming from cells in the nucleus accumbens. The major cell type in the accumbens are medium spiny neurons, D1 subtype of medium spiny neurons. Many of them express dynorphin. So Matt got a dynorphin-cree animal, so he could express transgenes in dynorphin-producing neurons. And then he expressed tetanus toxin in the nucleus accumbens, a Cree-dependent tetanus toxin. And most of you know tetanus toxin cleaves a critical protein involved in transmitter release and silences neurons. It prevents transmitter modulator release. And to our surprise, this manipulation had no effects on the sociability deficits. And we did some positive controls. We did some slice physiology to convince ourselves that tetanus toxin was working. So this suggests that the norphin that is presumptively being released to activate the kappa receptors in the accumbens is not coming from medium spiny neurons. Where might it be coming from? Matt looked at the Allen Brain Atlas, did a lot of work, and it turns out there's a population of dynorphin neurons in the dorsal raphe nucleus that express dynorphin. So we reason, and they send projections into the nucleus accumbens. So we reason maybe this is the source of dynorphin that is activating the kappa receptors during withdrawal that might be necessary for the sociability deficits. And in fact, when he expressed tetanus toxin in dynorphin expressing neurons in the dorsal raphe nucleus, he got a profound and robust block of the sociability deficits during opioid withdrawal. So this suggested the hypothesis that for whatever reason, these neurons are getting activated by the morphine or during the withdrawal, they're releasing dynorphin in the accumbens, and that is necessary and perhaps sufficient for the sociability deficits. So the next experiment he did was, what if he optogenetically releases dynorphin in the nucleus accumbens? What will happen? That's what the neurons look like in the raphe. So now he's expressing channelrhodopsin in dynorphin expressing neurons in the dorsal raphe, and activating the dynorphin expressing inputs in the accumbens. And remember, when we did this with serotonin inputs, we got a robust enhancement of sociability. Now he's getting a robust deficit in sociability. And again, the results are not subtle. Okay, so then is the dynorphin actually activating kappa receptors in the nucleus accumbens to mediate the sociability deficits? So again, the beauty of optogenetics is what he can do is in all the animals, he can activate the dynorphin inputs by activating the cell bodies. He can then turn off the light and get their normal sociability. Then in half the animals, he can infuse saline into the accumbens and then activate the dynorphin neurons. And in the other animals, he can infuse a kappa receptor antagonist into the accumbens. And what you can see is that infusion of a kappa receptor antagonist in the accumbens prevents the sociability deficits elicited by, I know it's a lot of words here, dynorphin input activation in the accumbens. Even I saying that, it's a lot of words. It's all going to come together. So then we wanted to know where are the kappa receptors localized. What I forgot to mention is the other reason we were interested in kappa receptors from the very beginning is there was already a literature reporting that serotonin neurons express kappa receptors, that dopamine neurons that send inputs in the accumbens express kappa receptors, that kappa receptor activation on terminals or inputs from dopamine and serotonin inputs reduces dopamine release, reduces serotonin release. So hopefully this is coming together. The hypothesis is maybe kappa receptor activation on serotonin inputs in the accumbens is reducing serotonin release and contributing to these sociability deficits. But there are kappa receptors also at other places. So we took a genetic approach. We got a kappa receptor conditional knockout mouse. Matt first put a Cree virus into the accumbens to, in theory, genetically delete all the kappa receptors in the endogenous neurons in the accumbens, the median spiny neurons. That had no effect on the sociability deficits. He then crossed the kappa receptor conditional knockout with a DAT-Cree line. So he's deleting the kappa receptors only from dopamine neurons. Those animals still showed the sociability deficits during protracted withdrawal. He then crossed the conditional kappa receptor knockout with the Circuitry line, deleting it from serotonin neurons. And obviously that prevented the sociability deficits during protracted opioid withdrawal. So we now have a clear hypothesis. During protracted opioid withdrawal, dynorphin is being released in the accumbens, activating kappa receptors on serotonin inputs, reducing serotonin release that we believe is necessary for normal pro-social interactions. So by this time in the project, there are Yulong Li and others in the field have developed genetically encoded sensors for measuring neuromodulators and changes in their levels. So one of these is called GRAB serotonin. It's based on a serotonin receptor. So you take a virus, you express the GRAB serotonin in the nucleus accumbens. When serotonin is released, it binds to this exogenous artificial receptor and it fluoresces. And what this shows is if we put GRAB serotonin in the accumbens and then we do fiber photometry, so we're measuring bulk fluorescence, and we give the animals MDMA. Remember I said MDMA is well known to cause massive release of serotonin. We get a massive fluorescence change in the nucleus accumbens, just providing more, I would say, more definitive evidence that MDMA causes serotonin release in a very robust fashion. So now what Matt can do, it's frozen again. There we go. All right. You're not going to hear about mouse empathy. I'm going to finish it before I talk about that. He's now doing this experiment I showed you earlier in the talk. It's a little bit different. So now he's measuring serotonin levels in the accumbens using GRAB serotonin during a social interaction. Our subject mouse, time zero is again, as shown in this little cartoon, is when our subject mouse is just approaching the little juvenile. And you can see that you do get a modest but clear enhancement in GRAB serotonin fluorescence, meaning an increase in serotonin release, which is exactly what you would expect, because I previously showed you that our GCAMP measurements measuring the soma activity of serotonin neurons, we saw a graph that looks pretty similar. So then half the animals got saline, half the animals got morphine, went through withdrawal. And what you see, it's noisy data, is that the saline treated animals, when tested for sociability a month later, still show the increase in serotonin in the accumbens, but the animals going through protracted opioid withdrawal do not, on average. So is that lack of increase in serotonin levels in the accumbens during a social interaction, does it require the Kappa receptors? So now he's doing the same experiment, but now all the animals are going through the morphine administration and withdrawal. Half of the animals are wild type, half of them are mice in which we've genetically deleted Kappa receptors from serotonin neurons. So this is the baseline measurements of serotonin, baseline sociability. And what you can see is all of these animals have gone through morphine administration and opiate withdrawal. The animals, the wild type animals, show the lack of increase in serotonin. The knockout animals show a roughly normal increase in serotonin. And then finally, last data slide, I think, because I'm not going to tell you about mouse empathy, I'll spare you that, is we then did the same experiment we had done in the autism mouse model. So now we're expressing GCAMP in serotonin neurons in the dorsal raphe nucleus and looking at baseline sociability and then the sociability deficits. Looking at the animals during their protracted opiate withdrawal. So these animals show the sociability deficits, but you can see that the activity of the serotonin neurons themselves is completely normal. Yet, so the hypothesis is that, well, let me just conclude. I think it's in, if this computer will allow me to. And then I am going to wrap up and spare you the rest of this. Yeah, yeah, no, I know. Oh, whoops, what do I do here? Thank you for how do we escape out of here? Yeah, end show maybe. All right, I'm gonna wrap up. Let me find where I am. I only had like 60 slides. It was an hour talk. Here we go. Oh no, I have one more data slide, then I'll wrap up. Hey, I drove a long way to be here. So does this have any potential clinical relevance? A reviewer, the paper just got accepted to Neuron, and a reviewer appropriately asked us, what about if we give a Kappa receptor antagonist that is in clinical use? This is the same drug that Andrew Crystal tested in his anhedonic patients. And what this shows is that if we give the animals this drug aticaprant, which is a Kappa receptor antagonist, and this is a crossover design, you know, it was a well-controlled behavioral study. You can see that giving animals aticaprant, you know, I think it's about an hour before the sociability testing, largely reversed the sociability deficits. So the summary of this, then I'll wrap up, is that the sociability deficits during withdrawal, opioid withdrawal, require dynorphin release in the accumbens from an unexpected population of dynorphin neurons. This dynorphin activates Kappa receptors on these serotonin inputs to decrease the serotonin release. That results in social deficits. And that what I try to convince you of is that the sociability deficits in two different mouse models of prominent neuropsychiatric disorders, a genetically based autism mouse model and a sociability deficit in protracted opioid withdrawal, both involve decreases in release of serotonin in the nucleus accumbens, but via very different mechanisms. And then finally, this is a slide, the last point is, unfortunately, the brain is really complicated because if my results are correct, and I always say, I've given this talk in public, I wish people would try to replicate these results. We need replicability. You should not trust anything I say until you do the experiment yourself. I'm exaggerating a little bit, but please. But this says that this complex neuromodulator serotonin is itself modulated by other neuropeptides. And there's a lot of examples of that, but it just says, things are really complicated. So I'm going to skip the empathy in mice. We had a paper in Science about a certain form of empathy. It's called the social transfer of pain and analgesia. It's actually a pretty cool thing to study. The only thing I'll tell you is this one slide, and then I will wrap up. So the model is the following. You take two mice that have hung out with each other, and it's important that it helps if they've hung out with each other previously. It works if they haven't, but you take cage mice. One mouse you put in pain. Doesn't matter the source of the pain. You then let that mouse have its pain for a little while. You then take a bystander observer mouse and you let it hang out with the mouse in pain for an hour. It actually can be done in less time. And then you test the pain responsiveness of the bystander mouse. And the bystander mouse shows pain behaviors just from hanging out with another mouse in pain. This is a very well-established behavioral model now. The Science paper, we did a lot of experiments on this to convince us. And what this is just showing you is that, so this is the mechanical threshold. So the lower the threshold, the animal is basically in pain. It's a paw withdrawal assay. So the animals that actually were given pain in their hind paws show a pretty low mechanical threshold. They're in pain. Here are the saline bystander mice. They show a little bit of decrease in their threshold, but not a lot. This is the key. These are the mice, the bystander mice that got MDMA. So we gave them MDMA immediately, like 10 minutes before we let them hang out with the mouse in pain. And for reasons, I have no idea what the mechanism is, although we have ideas about it. This says that the MDMA experience made this bystander observer mouse more sensitive to the affected experience of its little buddy. And we actually have the same results with the social transfer of analgesia. So finally, I didn't do any of this work myself. These are the postdocs who generated all the results. I emphasize, I don't have one of those slides with my 100 ex-postdocs and graduate students to show off how old I am. I don't need that anymore. But Jess Walsh did the autism, serotonin work, Boris Heifetz, MDMA, et cetera, et cetera. And finally, again, I want to end by thanking you, Sam, for giving me the opportunity to start my career, letting me hang out at UCSF for 10 years. I also want to thank Roger, who's sitting in the front row of the audience, for teaching me how to be a scientist. And like I said, you guys can decide whether to share the credit or the blame. And thank you all for your patience. Oops, I can check my email. How do I get off this screen?
I thought Rob would give a great talk, and one of its effects is that I'm really full of serotonin, baby. It was great. So I remember Rob well, and I remember he told the stories not exactly correctly, but we were LTP schmelTP. I said, but there were addenda. It's just great. I have 100 things I want to say, but the most important thing is that, thank you. I'm so glad we spotted you and that Roger nurtured you, and that we've lived in this wonderful community of scientists together, we and so many others here, for 35 years. And that although your goal and my goal and Matt's goal and many other people's goals is to take this stuff and actually make a difference in people's lives, because people are suffering so much. It's hard. It's really hard. But I must tell you that if I were asked 35 years ago that whether we would be at this stage, which is really, in my view, rather promising, I would be very surprised. the amount of advance that has happened, and it's come from it's come from everything, and it accelerates, and it's so there's hope, and we got to keep trying, and the suffering is not going to go away. And anyway, thanks a lot.
Coming through on the next.
Sure, we're going to take a few questions now. And yeah, please use the microphone.
So I'm Gray Davis, UCSF. I'm struck by something that we're restricted about as experimentalists, which is the acute perturbations, with the exception of some of the knockouts and serotonin grab that you were talking about. But one of the things that seems to be the flip side of what you were talking about is the general tone of serotonin or octopamine in the nervous system, and what is setting the behavioral states of the organism. And do you have any insights on how we might get there?
No. For a variety of methodological reasons, we're not comfortable like trying to use these genetically encoded sensors to make any estimates about changes in, as you say, tone or basal levels of serotonin. Not really. I'm just thinking mechanistically. I'd have to think the ambient environment of neuromodulators in a structure like the accumbens, right? It's going to be influenced by basal levels of activity in the somas, by levels of transporters, all those complexities. So no, I don't have any brilliant ideas of how to address that issue. And I don't have any strong opinions on how important it is or not, to be honest. I just don't know. I think it's remarkable. Again, Sam gave the optimistic. I'll try to stay optimistic that we've been studying dopamine now for, 70 years. And I'm changing the subject of it. I'm getting on my soapbox, which I like to do. One of the most robust behavioral results in the rodent world, especially since the development of optogenetics, is if you release dopamine in target structures, like the specific target structures, like the ventral striatum, also the dorsal striatum, the animals experience that as highly reinforcing. They will press a bar forever. And you can do that with a drug of abuse and in my view, the field has no idea how that actually really works. There are primitive hypotheses about dopamine influencing activity of different populations of neurons in these structures. But if you actually look at the data and think of it, we just don't have an understanding. So the point I'm making is you're adding even the additional complexity on that as how to ambient levels. It's just going to be brutally, that complexity I don't want to deal with yet. I think the first question is, can you figure out what dopamine is doing in a specific target structure like the accumbens to ensemble activity? That's where I'm becoming a systems neuroscientist, because I'm going to have to say in codes pretty soon. And compare that to the release of serotonin, which are experiments we're doing right now, because behaviorally, we know these modulators, when we manipulate them optogenetically, have clearly different behavioral effects. And so I think right there, that would be an important contribution to the field. And so that's the direction we're kind of headed. And we're also developing with a colleague near Eshel, who's a computational dopamine person who I am slowly getting him to think more mechanistically. We're developing hopefully more sophisticated behavioral assays to start addressing some of these questions. Anyhow, blah, blah, blah.
Okay, we have a question from Howard Fields.
Howard.
And he says you gave a wonderful talk.
Thank you, Howard.
Any experiments with ketamine? It's supposed to be an NMDA receptor antagonist, and it does produce robust but short-lived remission of depressive symptoms. Do you think ketamine affects the serotonin system in depressed patients?
It's a good question. I have no strong opinion on that, Howard. I've very intentionally never done a ketamine experiment in my lab. I did write a commentary with Carl Deiseroth in Nature a few years ago, where I made the same argument that if we really want to understand how ketamine might be therapeutic in the treatment of depression and perhaps other neuropsychiatric disorders, we need to first figure out where in the brain it's acting, which cell types, its molecular targets. And the truth is, there's a little controversy about its molecular targets. There is, whether it's influencing serotonin, hasn't been looked at. There's a paper in Neuron from Genia Kozlovetsky, I forget how to pronounce her name, at Northwestern, reporting that ketamine actually influences dopamine neuron, one of your VTA dopamine neuron activity, and its inputs into prefrontal cortex. But in terms of serotonin, I don't know, and I'm steering away from that. I'm focusing on serotonin drugs, Mdma and psilocybin, with some colleagues here at UCSF.
I think we have time for one more from Marty Horowitz.
Marty, how are you, Marty? Marty was down the hall from me on the third floor of Langley Porter in the 90s. So nice to hear from you, Marty.
All right, Marty asks, do you think these circuitry synaptic strengths are the way unconscious self other schemas work, producing predictive codings for present moment social situations?
Yes, I do. How, because I believe the brain and its complex neural activity mediates everything, including our unconscious motivations, our unconscious perceptions, our unconscious biases. So by definition, I have to believe that there are complex, activities of millions of ensembles of neurons that are telling me. I have fond memories of you, Marty, and my projections onto you are only positive. All right, so thank you very much.